Welcome to this video on monosaccharides, structure and functions. Monosaccharides are the simplest unit of carbohydrates, which are the most abundant biomolecules in nature. Chemically, monosaccharides are defined as polyhydroxyaldehydes or polyhydroxyketones. By the end of this video, you should be able to understand nomenclature, classification, and important concepts like stereochemistry, anomers, epimers, optical isomers, mutarotation, and the biological significance of monosaccharides. Let us discuss classification. Based on the structural complexity, they are divided into three major classes, including monosaccharides, disaccharides, and polysaccharides. Saccharide is a Greek word which means sweet. Therefore, because of their sweet taste, they are named as saccharides. In this video, I will focus on the monosaccharides only. Let me present overview of monosaccharides. They are the smallest units of carbohydrates, also known as simple sugars. Generally, they are sweet in taste and water soluble. The smallest monosaccharides are trioses, which are made up of 3 carbon. Examples are glyceraldehyde and dihydroxyacetone. The largest monosaccharide unit is made up of 7 carbon. Examples are heptose, heptulose or pseudoheptulose. Chemically, they are defined as polyhydroxyaldehydes or polyhydroxyketones. Let us explore the chemical structure of monosaccharides, or in other words, why they are called polyhydroxyaldehydes or polyhydroxyketones. In the beginning, many of these carbohydrates, they showed empirical formula CH2O, and therefore they were called as hydrated carbons, but many of them later did not conform to this, and therefore the definition was altered. And the reason was because they have consistently multiple OH group and at least one ketonic group or one aldehydic group. Therefore, chemically, they are defined as polyhydroxyaldehyde or polyhydroxyketone. So let us explore the nomenclature of monosaccharides or naming of monosaccharides. The naming of monosaccharides is based on 4 criteria. The first one is number of carbon atom. Number 2 is presence of key functional group. Third is stereoisomerism, and the last one is optical properties. I will discuss these in detail in the coming slides. Let us explore the classification based on the number of carbon atom. According to this scheme, count the number of carbon atom, put a corresponding Greek word for that number, followed by OSE as the common suffix. Therefore, monosaccharides with 3, 4, 5 or 6 carbon atoms will be named as triose, tetrose, pentose and hexose respectively. Let me introduce some common monosaccharides about which you will hear during your classes on carbohydrate metabolism in biology and biochemistry. Important trioses are glyceraldehyde and dihydroxyacetone. Pentoses are ribose and deoxyribose. Glucose, fructose and galactose are common names in hexoses, and heptoses include pseudoheptulose. Let us explore the classification based on the type of functional group. A monosaccharide may have a keto or an aldehyde as a functional group. According to this scheme, put aldo or keto as the prefix followed by counting the number of carbon atom and put the corresponding Greek word for that number followed by OSE as the common suffix in the end. According to this scheme, this compound has 6 carbon and 1 aldehyde group. Therefore, this will be called as aldohexose. This second compound has a keto group and with 6 carbon atom. Therefore, this will be a ketohexose. Using the same scheme, you can easily name any monosaccharide. Now I will focus on the stereochemistry of monosaccharides. This is an important concept and topic in organic chemistry, including monosaccharides. In this topic, I will discuss stereoisomerism and stereoisomers, basis of stereoisomerism, and nomenclature of stereoisomers. Let us examine the concept behind stereoisomerism and stereoisomers. Stereoisomerism is the property of having different structures for a given chemical formula formed by special arrangement of group around one or more chiral carbon atom. Such structures are known as stereoisomers. Therefore, to understand the concept of stereoisomers or stereoisomerism, let me explain the concept of chiral carbon atom with the help of this 4-carbon compound having an OH group attached to third carbon. Except for the carbon number 3, every other carbon in this molecule has 2 or more than 2 hydrogen atoms attached to it. Such carbon atom is called as symmetrical or achiral carbon atom, and such carbon atoms are not going to form alternative structure. On the other hand, if you look at the carbon number 3, it has 4 different group attached to it. First group is ethyl group, second is methyl group, third is hydrogen, and fourth is OH group. A carbon atom attached to 4 different groups is known as asymmetrical or chiral carbon atom. This is also known as chiral center, and this carbon has a capacity to form alternative structure. In this molecule, OH is right now on the right hand side, but in alternative structure the OH may be facing toward the left hand side. Such structures around one chiral carbon atom are known as stereoisomers. So stereoisomers are the product of chiral carbon or chiral center. Therefore, to understand the concept of stereoisomerism or stereoisomers, you must be able to identify the chiral carbon in a given monosaccharide. Let us explore the naming of stereoisomers with the help of glyceraldehyde, the smallest monosaccharide, which is used as a reference molecule to name any other stereoisomers. First task, as I said earlier, is to identify the chiral carbon atom in this molecule. You have 3 carbon atom in this molecule. Carbon 1 and carbon number 3 are symmetrical, whereas carbon 2 is asymmetrical carbon atom because this is attached to 4 different group: aldehyde, methoxy, hydrogen and OH group. And therefore this chiral center can form alternative structure by orienting OH group toward the right hand side. So there is one single chiral center, which is carbon atom number 2 in this molecule. On a single chiral carbon center you can have 2 alternative stereoisomers. So this is very important point. Single chiral center makes 2 isomers. How they are named: when OH is facing toward left hand side, that isoform is named as capital L or LIGO. When it is facing toward the right hand side, it is known as capital D or Dexter isoform. How this molecule is used as a reference to name other stereoisomers, this is very important point to note. Other stereoisomers are named by comparing orientation of the chiral carbon atom equivalent to the chiral center or chiral carbon atom of glyceraldehyde, which is carbon atom number 2 in this molecule. Let us explore the naming of higher stereoisomers such as tetrose, pentose and hexose. As I said earlier, higher stereoisomers are named by comparing the orientation of their chiral center with that of glyceraldehyde. In these molecules, boxed chiral centers are equivalent to that of glyceraldehyde. Since these reference chiral centers in these molecules are farthest from their aldehyde group, this carbon is also known as farthest chiral center or farthest chiral carbon. Once you identify the reference chiral center, name as capital D or capital L depending on the position of OH group on the right or on the left hand side respectively. Since in all these molecules OH group is on the right hand side, they are named as capital D stereoisomers. So D-aldotriose, D-aldotetrose, D-aldopentose and D-aldohexose. To simplify, identify the orientation of the farthest chiral center to name a stereoisomer. To test your knowledge on the chirality, there is a query here, questions: how many stereoisomers are possible for the following dihydroxyacetone? You have to identify the chiral center if there is any. The answer is none. Because this is a symmetrical compound, there is no chiral center and therefore there is no stereoisomers. Let us calculate the number of stereoisomers for a given monosaccharides such as triose, tetrose, pentose and hexose. To do that, identify the number of chiral center in these given molecules. To make it easier, except for the first carbon in all these molecules is symmetrical and the last carbon is also symmetrical. So except for the first and the last carbon atom, remaining all other carbon atoms are chiral centers. And since a chiral center produces two stereoisomers, the total number of stereoisomers can be calculated using the formula 2 raised to the power n. N is chiral centers. So for triose or like glyceraldehyde, you can have two stereoisomers. For tetrose, there are two chiral centers. You will have 4. In pentose, you have 8. And in hexose, you will have 16 different structural stereoisoforms. There is a query again on stereoisomerism. How many stereoisomers are possible for the following hexose, which is fructose? So to answer that, you have to identify number of chiral centers in this molecule. Carbon number 1, 2 and 6, these are symmetrical carbon. The remaining carbon number 3, 4 and 5, they are chiral centers. So there are 3 chiral centers. So put the value in the formula 2 raised to the power n, which comes out to be 8. So answer is 8. You take a pair of capital D, capital L isomers. They are mirror images of each other. For example, this is an example of hexose. D is mirror image of L isomers. They are laterally inverted. They are non-superimposable. Similarly, in case of aldotriose also, D, capital D is mirror image of capital L. They are non-superimposable. Capital D and capital R, L isomers are also known as enantiomers. Only D isomers or stereoisomers are biologically active. So in summary, stereoisomers are alternative structure for a given chemical formula. They are named as capital D, capital L according to the orientation of the OH group on the farthest chiral center. In aldohexoses, except for the first and the last carbon atom, all remaining carbon atoms are chiral centers. Each chiral center can form two isomers, therefore the number of stereoisomers can be calculated using the formula 2 raised to the power n. N is the number of chiral centers in the molecule. Capital D and capital L isomers are mirror images. They are also called as enantiomers. Only D isomers are biologically active. Let us examine anomeric carbon and anomers. I will now focus on a special type of stereoisomers called anomers, which are formed by the orientation of the OH group on the anomeric carbon of monosaccharides. To clearly therefore understand the concept, I will first explain the anomeric carbon and then examine the anomers and their naming using glucose and fructose as separate examples. So let me further explain the anomeric carbon atom and orientation of OH group on this carbon atom forming anomers or isomers. This is glucose structure. Carbon 1 is carbonyl carbon atom. As I mentioned earlier, it has a tendency to form hemiacetal ring with OH group on the carbon 5. And as a result of this cyclization, there is a ring formed which is known as pyrene ring. In this pyrene ring, carbon atom 1 now is a new chiral center. This is derived from carbonyl carbon atom and therefore this is known as anomeric carbon atom. Orientation of OH group on this anomeric carbon atom forms cyclic isomers which are known as anomers. We will identify in next slides the types of anomers and how they are named. Similarly, in fructose also the carbonyl carbon atom which is symmetrical at carbon position number 2 has a tendency to make hemiketal ring structure with OH group of the carbon 5, and this structure is planar structure heterocyclic 5-membered ring, In this cyclic structure, the carbon 2 is the anomeric carbon atom. The anomeric carbon atom is derived from a symmetrical carbonyl carbon atom. So you should be able to differentiate between the carbonyl carbon atom and anomeric carbon atom. Orientation of OH group on this anomeric carbon atom will form cyclic isomers. They are known as anomers. We will discuss these in the next slide. Before describing types of anomers and their nomenclature, I would like to go over the correspondence between Fischer and Haworth projections. You should know that Fischer projected D-monosaccharides in the form of a linear structure, whereas Haworth projected them in a corresponding ring-like structure or planar ring-like structure. The question is, how are the OH group in the linear structures projected in the ring structure or Haworth ring structure? There are two rules to govern that. First rule says write down the groups which are present on the right hand side, such as carbon 2 and carbon 4. They will face below the plane of the ring. Second rule says left up. The group which are present on the left hand side, such as carbon 3, will face above the plane of the ring. Carbon 5 also has OH group projecting toward the right hand side, but it is not shown in the ring structure because it has been utilized in the formation of this hemi-acetal linkage in case of glucose or aldoses, and hemi-ketal in case of fructose and ketoses. Same rules will apply to all other monosaccharides including aldoses and ketoses. So this is the way OH groups are oriented in the linear in the ring structure. Now I will discuss the anomers and their nomenclature in the upcoming slides. Let us discuss the types of anomers and their nomenclature using example of glucose. This is the hemiacetal ring for glucose, where carbon 1 is a chiral center. This is known as anomeric carbon atom. Orientation of OH group on this anomeric carbon atom will define the type of anomers and their name. When OH is facing downward or below the plane of the ring, or in trans with 6CH2OH group, that isoform is known as alpha anomer or alpha D glucose or alpha glucose. Similarly, when OH is facing above the plane on anomeric carbon atom number 1 or in cis with 6CH2OH group, that isoform is known as beta anomer and beta D glucose in this case. Let me explain the types of anomers and their nomenclature using fructose as an example. Similar to glucose, there is a ring structure in fructose, but it is slightly different. This is known as hemi-ketal ring structure, in which carbon atom 2 is a chiral carbon atom, which is known as anomeric carbon atom. So the position of OH group or orientation of OH group on chiral carbon number 2 or anomeric carbon 2 will define the anomers of fructose. When OH is facing downward or in trans with 6CH2OH, that isoform is known as alpha anomer or alpha D fructose. Similarly, when OH is facing up above the plane of the ring or in cis with 6CH2OH group, the isoform is known as beta anomer or beta D fructose. So this is how the anomers are classified and named. We have discussed chirality, types and nomenclature of stereoisomers so far. Now I will discuss their optical properties, types of optical isomers and their nomenclature. Stereoisomers can rotate the plane of polarized light to the right hand side or to the left hand side. Before I discuss these in detail, I would like to explain briefly the polarized light. In normal light or unpolarized light, the vibrations occurs in many planes perpendicular to the direction of propagation. However, in plane polarized light, vibration occur in a single plane oscillating perpendicular to the direction of propagation. Monosaccharide solution can rotate the plane of polarized light in a clockwise or anti-clockwise direction due to presence of chiral carbon, forming two types of optical isomers. We will discuss these and their naming in the next slide. So let us discuss optical isomers. Optical isomer is a property or function of chirality in the molecule. Therefore, any stereoisomers with a chiral carbon atom can rotate the plane of polarized light in a clockwise or right-handed manner. Such isomers are known as d isomer, small d isomers, or plus isomer or dextrorotatory. They can also rotate the plane polarized light in an anti-clockwise or left-handed manner, and such isomers are known as small l or levorotatory isomers or minus isomer. The notations d and l or plus and minus for optical isomers are different than capital D and capital L isomers. Let us discuss mutarotation. We have learned that optical rotation is a function of the chirality of stereoisomers. However, optical rotation of anomers changes gradually over time until it reaches a constant value. This phenomenon is known as mutarotation. For example, a freshly prepared glucose solution shows 112 degree clockwise rotation, whereas beta D glucose shows 18-degree clockwise optical rotation due to interconversion of the ether rings through a linear open chain structure. This leads to the establishment of an equilibrium mixture of 36% of alpha D and 64% of beta D glucose or beta D anomers, producing 53-degree clockwise optical rotation. This is an example of mutarotation in glucose. Other anomeric pairs of the monosaccharides such as fructose and galactose also undergo mutarotation. Therefore, mutarotation is a characteristic feature of anomers. So let us review the summary of anomers. Carbonyl carbon 1 in aldoses such as glucose and carbonyl carbon 2 in ketoses such as fructose form the anomeric carbon, giving rise to two cyclic isomers which are known as anomers. Alpha anomer has the OH group orienting below the plane of the ring, whereas beta anomers has OH group projecting above the plane of the ring. Optical rotation of an anomeric sugar solution changes on standing due to their interconversion of alpha and beta anomers. This phenomenon is known as mutarotation. Equilibrium mutarotation value is a characteristic of a monosaccharide. Therefore, deviation from the characteristic mutarotation may indicate impurity. So, this is the significance of mutarotation. Let us discuss epimeric carbon and epimers. Epimers are stereoisomers that are formed by the configuration of the OH group on the epimeric carbon. To understand the epimers, therefore, we should define the epimeric carbon first. Let us define epimeric carbon and epimers. An epimeric carbon must be a chiral carbon. In glucose, carbon 1 and carbon 6 are achiral or symmetrical, while carbon 5 defines D and L stereoisomers, so they are not the epimeric carbon atom. In glucose, however, chiral carbon number 2, 3 or 4 can act as epimeric carbon. Two monosaccharides or a pair of monosaccharides that differ in configuration at only one such chiral carbon are called epimers. Epimers therefore are diastereomers. They are not mirror images or enantiomers. In the next slide, to make the concept little more clear, I will present examples of epimers. So here is the first example of epimers that includes glucose and galactose. Alpha D glucose and alpha D galactose are epimer with respect to the orientation of OH group on carbon number 4. Remaining structure as well as the orientation of the OH group are identical in both molecules. These are not enantiomers or mirror images, they are diastemers. Here goes the second example of epimers that includes glucose and mannose. Alpha D glucose alpha and alpha D mannose are epimers with respect to orientation of OH group on carbon atom number 2. The remaining structure as well as orientation of the OH groups in both molecules are identical. Again, they are not enantiomer, they are not mirror image, they are diastereomers. There is a query: how do epimers differ from enantiomers? The answer is epimers are diastereomers, they are not mirror images, while enantiomers are mirror images. Let us discuss reducing property of sugars. Any carbohydrate, including the monosaccharides such as glucose, fructose, and galactose, which has a reactive carbonyl group such as aldehyde, CHO group, or carbonyl group, is considered a reducing sugar. This property has been exploited in developing a diagnostic test such as Benedict test, which is used for measuring glucose level in the urine of diabetic patients. In the next slide, I will discuss the principle of the Benedict test. Let me explain the underlying principle of the Benedict test. Glucose solution in alkaline pH reduces cupric ion to cuprous, forming a red brick color, which is an indicator of presence of glucose in the urine of or biological sample. Whereas aldehyde group is oxidized to carboxylic group forming gluconate, and this happens through the formation of an intermediate known as anidiol. So this is the test which has been developed utilizing the reducing property of glucose. Let me start with the significance of these biomolecules. They are vital from both physiological and biochemical perspectives. They serve as primary energy sources and storage molecules such as glucose, starch, and glycogen. They also form key structural elements in biomolecules, cell membranes, and cell walls. In addition, they also help in differentiating the self from the non-self and defining the blood group antigens. Failure to utilize or metabolize carbohydrate properly leads to diseases such as diabetes, galactosemia, fructosuria, lactose intolerance, and glycogen storage diseases. Carbohydrates are sweet in taste, therefore let us present sweetness with respect to sucrose, which is a standard reference sweetener. If you see the data here, fructose appears to be the sweetest at 175% compared to sucrose, which is taken here as 100% reference point. Glucose is at 75%, followed by maltose 33%, galactose 30%, and lactose at 16% compared to sucrose. Therefore, fructose appears to be the most or sweetest monosaccharide. So in summary, monosaccharides are defined as polyhydroxyaldehydes or polyhydroxyketone. We discussed their classification based on the number of carbon atom. They were defined as trioses, tetoses, pentoses, hexoses, or heptoses. They were also defined based on the functional groups such as aldoses and ketoses. And we also define based on the stereoisomers as capital D, as capital L, or based on the OH group orientation at the farthest chiral carbon atom. We also define them as alpha and beta anomers based on the orientation of OH group on the anomeric carbon atom. And we also define them based on their optical properties as small d and small l, dextrorotatory or levorotatory. And finally, we define them by the configuration of epimeric carbon atom. So this is the end of the monosaccharides. Thank you very much for watching this video. Please like it, share it, and subscribe it. The other part of carbohydrate such as disaccharide and polysaccharides will be presented as a separate videos later. Until next time, you take care. Bye bye.